Afternoon everyone. I'm just going to give you a brief story of my PhD project. So the project focused on Tribolium castaneum, which is also known as the rust red flower beetle. There's an image of it there. And essentially this beetle is a major pest on stored cereal grains around the world, but we were interested into its infestation on stored cotton seeds in southeast Queensland. And the reason why we're interested in this is because upon closer inspection, the beetles aren't actually feeding on those seeds, even though we're getting numbers of millions plus beetles there. Instead, they're actually feeding on the fungal species on those seeds. So we wanted to know what this meant in terms of their ecology outside of grains, and if we could perhaps use these fungal species in terms of attraction for a pheromone lure or something like that. So there were three main aspects of the project. Firstly, identifying the different fungal species on those cotton seeds. We had no idea at the beginning. I set out, identified those different species. I then thought, well, can we actually rear the insects on each of these fungal species? We tried all of them that we identified, and we could, but there was big variances depending on which fungal species it was. Then we thought, well, what's going on in terms of attraction? Is one of these fungal species drawing them in to the cotton seeds? Tested that in the lab. We did olfactometer tests, wind tunnel experiments, and we also did some field experiments. That's this image here, where we set out some traps all around one of those sites in the field and saw what went on. What we found was out of a dozen or so different fungal species that we tested, a few of them were really pulling them in and a couple were actually repelling them. We then moved on to looking at what specific volatiles each of those fungal species produce. The idea here is that once you ID the volatiles, perhaps you can up the concentration and look at making a lure or some sort of repellent agent. And that's why I went to number 3 here, which is where we took those fungal species, looked at their volatiles using gas chromatography and mass spectrometry machines, analysed them, got a list of what volatiles were there, obtained each of those separate chemical compounds, and then moved on to trials in the lab looking at the insect's response to each of those compounds. Overall, what we've found is that fungal species across the agricultural landscape probably are playing a big part in this species ecology, specifically outside of stored grains. And perhaps if we continue more research into those specific volatiles, we could be looking at the use of a product or either to attract them or to repel them. But yeah, thanks very much. That's a brief overview of the project. Cheers.